very excited to be able to welcome Dr. Haitham Elmarakeby, who is an instructor at the Dana-Farber Cancer Institute, as well as the Harvard Medical School, and is also an affiliate researcher at the Broad Institute of MIT and Harvard. His research focuses on using machine learning to better understand cancer progression and therapeutic resistance in cancer patients. And right now, he's currently focusing on building interpretable models to discover novel markers of clinical and biological outcomes in multiple different cancer types. Haitham earned a bachelor's degree in systems and computer sciences at Al Azhar University in Cairo, and received his master's in computer engineering at Cairo University. After that, he completed his PhD in computer science at Virginia Tech and has been a postdoc at Dana-Farber after that. And we're excited to welcome Haitham here, and we're excited to hear about some of his latest and greatest research in prostate cancer discovery using biologically inspired networks. So Haitham, please take it away.
Yeah, thank you. Thank you for the introduction, and thank you for giving me the opportunity to present my research today. Thank you everyone for taking the time to attend the seminar today. So today I'll be talking about customizing neural networks to better understand cancer progression in prostate cancer. So let's get started. So the current understanding of cancer is that it is a genetic disease, which means that mutations in the normal cells play a major role in deciding the fate of the cancer cells. So if you imagine like a normal cell that acquires a somatic mutation, probably due to random change or due to external factor like exposure to chemicals or radiation, this first mutation can be passed through division to the offspring of the cell. And these offsprings, the newly developed cells may acquire more mutations. Some of them may acquire more mutations. And you can imagine that through this sequence of divisions, these cells may acquire like second, third, and fourth mutations. And through the accumulations of such somatic mutations and driver mutations in these cells, these cells may form a lesion somewhere in the body and may acquire some characteristics that helps these cells to grow and divide in uncontrolled way. So I am interested in understanding how these mutations affect the different outcomes in the patients. So I'm interested in getting all of these mutations in the patient and associating these mutations to different clinical outcomes, such as whether the patient has his tumor growing or shrinking, whether the patient is responding in a proper way to the current treatment, or even predicting the survival of the patients. So associating the whole genomic profile of a certain patient to a certain clinical outcome is a very interesting research area, and it is a very challenging one as well. So one problem that happens is that when you start associating a single mutation to a certain clinical outcome, you will find it very difficult because the frequency rate of such mutation is very low in the population. So you can imagine that two patients may have similar phenotype. They may have the similar like cancer type maybe. However, they harbor very different genomic profile. This is because different mutations may have similar effect. So this is a major problem when you try to associate like individual mutations to a certain clinical outcome. So an actual way of thinking is that probably we can combine these mutations or aggregate the signals from different mutations using certain biological units in the genome. For example, aggregating these mutations on the gene level. With the assumption that maybe different mutations in the same gene may have similar effect. So even if you do this, so this is a representative study from our lab that is published in Nature Genetics 2018. We collected a lot of prostate cancer samples. We have the whole exome sequence for all of these samples, and we tried to see what genes are mutated in these samples. And with the exception of TP53, probably you will find that most of the genes are mutated with very low frequency. So it is like 1, 2, or 3% of the population, they have mutations in a certain gene. So we end up with very long tail of genes that, frankly, we don't know exactly what to do with these genes, or how to rank them in a reasonable way, or how to make sense of all of these genes. We have a lot of these genes that are mutated in very low frequency. So if we continue this theme of aggregating signals from different parts of the genome on a higher level biological entity, probably the next step is to think about pathways. So this is a representative study as well from TCGA consortium, where they suggested to use these 10 pathways to study different cancer types. With the assumption that if you have like mutation in one of the genes in a certain pathway, probably mutating one of these genes or the other they have the same effect as long as they are in the same pathway. So this is an extension of the theme of aggregating signal from the mutation level to the gene level and now to the pathway level. Here in our project, we are trying to use deep learning to solve the problem of associating like a certain genomic profile to a certain clinical outcome. And as we know, deep learning has achieved the state-of-the-art performance in so many domains and fields, including computer vision, speech recognition, natural language processing, and so on and so forth. And this success of deep learning can be attributed to three things. One is the advances in the computational elements, including CPUs, TPUs, GPUs, and even customized hardware accelerators of deep learning models. And the second thing is the availability of right now almost infinite number of samples to train your model on. So we have a lot of unstructured data and structured data in so many different forms and formats. And the third thing is the advances in the model development and the different techniques for training such models. So if you try to project these things on the cancer genomics space, starting with the computational elements, I don't think right now we have a lot of problems accessing high performance computing elements to train our models on. So with the HIPAA compliant services provided by different providers, and now you can train your model on the cloud without major problems. I would say that there are some logistic problems, but as long as you have enough money and you can pay for these things, you should be able to train your models on the cloud. When it comes to the data, we have a major problem in the field that we don't have the privilege of having access to like almost infinite number of samples. We have very limited number of samples to train our models on. And even worse, these samples are scattered everywhere. And you can imagine all of these healthcare facilities and hospitals, they are treating patients, sequencing their genomes, and storing this data in their own system. And sharing such data with each other is a major challenge in the field. To train any model, you need a lot of data, and to get a lot of data, you need to collect all of these samples from everywhere and try to run a data harmonization processing on this data before feeding this into your model. And this is why a major effort in our lab, which is mainly a computational lab, that we work with a lot of collaborators to collect a lot of data from everywhere and try to harmonize this data in a good way and publish this data and make it public for the community to work on and to run secondary analysis on. So this is one of our studies that we published on CBio portal. It is available right now. You can go and download it. So this is more than 1,000 patients from prostate cancer with all of the whole exome sequence data available. So you can go and see what genes are mutated and what are copy number events are there and things like that. And you can download it and run your preferred model on these data. When it comes to the modeling and the development of models, when you have a prediction task, probably you have a lot of options, and every option comes with trade-off between the accuracy and the interpretability. So if you imagine something simple like logistic regression, which is like just a bunch of weights, And you can train such model in a very fast way and get some results, but usually the accuracy is, or the computational performance is less than the other models. However, the interpretability of this model is very high, which means that you can inspect the model easily and get some sense of what feature is driving the outcome. However, if you'd like to use something like deep neural network, you may get like high performance, computational performance. However, the interpretability of such model is very bad, or I would say it is painful. So in this project, we are trying to partially solve this problem by enhancing the interpretability of deep neural network. So the goal here is to come up with a model that is more interpretable than the other models. So in cancer genomics, it is not enough for us as researchers to get some data, do some pre-processing, feed it into a model, and get some performance evaluations. Rather, we need more than that. We need to be able to open the black box and get some insights about what's going on. And the research in the interpretability methods and techniques has been very hot in the recent years. And there are a lot of techniques proposed out there, and there are a lot of themes in the attribution field and the interpretability field using attribution or visualization, things like that. So this is an example, for example, in visualizing different things that the model is trying to learn. So this is GoogleNet, and it is thought that the first layers of this model learns simple features like ages, and maybe the middle layers will learn something like parts of the images, and maybe the last layers will learn higher level representation of the inputs. So this is a kind of interpretation of the model where we are trying to learn what the model had learned and how it is working from inside. This is another way of interpreting the model by visualizing or laying down the importance score of different parts of the input space. So probably this is a classifier that is classifying the image as having a sheep, and you can see that there are some importance on the image of the sheep. So these are just some examples of the interpretability of different models, and I'll be talking more about this later. So in the next few slides, I'll be touching upon these things. I'll be talking about PNet. This is a model that we developed and published recently. And I'll be talking about the problem that we are trying to solve using this model, and some of the computational results and some of the interpretation. Hopefully, we'll come to some of the experimental validation of our findings. Just keep in mind that this is like we have a lot of feedback between different stages, and things do not go straightforward during the development. Rather, we have a lot of discussions and conversations regarding our results and our developed model. So if you imagine a simple model like that, where you have a bunch of inputs and you can aggregate all of these inputs using a bunch of weights, you can get the outcome as a function of the input by weighing these features differently. If you learn the weights W1 until W6, you will come up with a certain model. This model is either like logistic regression or linear regression based on the function f. This model is very simple. It is very fast. It is very interpretable. You can just inspect these weights and see which feature has high weight, and probably this feature has more importance than the other features. However, it may be too simplistic to assume that the process that you are modeling is a linear process where the input is the output is linear function in the input. If you like to have like more sophisticated model, probably you need more layers to define your model. So this is a network with more layers, and these are the connections between the layers. If you have a prediction task, probably you will start with something like that. Deep network. However, there is no guideline in the field about how many layers should you have, or how many nodes per layer you should have, or the kind of connections. So usually you start with arbitrary model and with some regularization, and probably randomly dropping some of these nodes using drop out, or dropping some of the connections using drop connect. you end up with some model that can fit your data. So as you can see, we have a lot of arbitrary things here. So we tried to do something more systemic. We tried to propose a model that is based on our prior knowledge about the hierarchy of biology. So we proposed a simple model where the inputs represent some kind of feature that you can measure. The next layer represents some kind of genes that you are interested in. And the next layer represents some kind of pathways that you would like to model into your model. And the next layer represents some kind of biological processes that are interesting for you as well. And the last layer is some kind of outcome. So this layer actually looks different. It is a sparse network. You have the input features, you have genes, and you have pathways, and you have processes represented into the model itself. The connection between these nodes are based on our known knowledge about how these things are connected to each other. So the connection between the features and the genes based on the fact that these features belong to this gene only. And the connection between the genes and the pathways based on our knowledge that these genes are a member of this pathway. And also the connection between the pathways and the biological processes are based on our knowledge that these pathways are part of this biological process. So as you can see, the idea is simple, is to control the flow of the information from the input to the output, associate certain meaning, some meaning to the layers, and give the nodes some meaning as well. So it seems simple, but in practice, you would like to include something like that into your network. So this is the set of pathways curated through the Reactome dataset. We have a lot of things here. We have some dots and some connections. Every dot here represents a certain pathway, and every connection here represents a child-parent relationship between this pathway and maybe higher level pathway, and so on and so forth, until you get like a biological process. And you have many of these biological processes as well. In total, you have like more than 3,000 pathways. So you'd like to include these 3,000 pathways into your network. And also you need to include the associated genes, the members of these pathways, and the feature that you are including as well. So in general, the PNet model will be like a big model. It's not a small one. However, it is based on this hierarchy of annotations. So what we did actually is that we took this abstract hierarchy, which is like curated by scientists over years, I would say decades, and transferred this into active model, a neural network model that can learn something and can change the weights to optimize certain loss function. So instead of having an arbitrary model on the right, we have a more structured and well-constrained model on the left. Instead of having dense model on the right, we have our PNet is a very sparse model. Instead of having middle layers that do not represent anything, we have some layers in our PNet that represent something. And also we have some meaning for the nodes as well. So as you may know, training a sparse network probably is more challenging than training a dense networks. And we found that empirically that adding like a classification layer after each layer in our PNet boosts the computational performance of our model a little bit. And these are the number of nodes per layer. And this is based on the Reactome dataset. If you sum all of these layers up, of all of these nodes up, you will end up with around 70,000 parameters in your network, which is a small number of weights if you compare it to other models. Doing so is important and useful in aggregating different data types. So if you imagine that you have multiple features, you can aggregate all of these features on the gene level automatically. You don't have to worry about how to combine these or how to assign weights for these features. Rather, the network will automatically assign these weights for you based on the loss function that you are trying to optimize. Also, it is our network is able to assign weights for different nodes. So you can go back and see what feature is affecting the outcome most, and what gene is affecting the outcome most, and what pathway and biological process are affecting the outcome most. And hopefully you can find a path from the input to the outcome that where these features and pathways and genes are maximum. So this is a very important thing to be able to assign importance scores for all of these biological entities. And probably this will help scientists and researchers to find or to nominate some biological hypotheses from these interpretations of the model. So the implementation of such things is not very challenging. Rather, if you think about like a dense layer, you can see the representation of a dense layer here, where you have like a vector X and vector outcome vector Y, and if you like, you can have Y as a function in the in X through multiplying by weight matrix W. If you have a sparse layer, if you like to include it, you can add a mask matrix here, M, and this represents the adjacency matrix between the two consecutive layers. And sometimes you have like a patterned sparse layer, like the first layer in our PNet. It is patterned, which means that there is a certain pattern in the adjacency matrix, and you can make use of this pattern to accelerate the computations a little bit. So next, I'll be talking about the problem that we are trying to solve here. So in general, we are interested in understanding how the cancer progress over time from the primary site to the metastatic site. The question that we are trying to answer here in this project is that whether there is any differences in the molecular profiles of the primary tumor compared to the metastatic tumor, and can we catch these differences, and can we know what are these differences are? So we started with the data that we published before. So the same data that we published before, we tried to do like a secondary analysis on it. And this data is, as I said before, it is more than 1,000 prostate cancer patients. We have the whole exome for these patients. 70% of these patients have their samples taken from a primary site, and 30% of the patients, they have their sample taken from the metastatic site. And for these patients, we have the mutation profile of the patients, all of the somatic mutations of these patients. We have the copy number amplifications and deletions. For some of them, we had gene expression, but we didn't include these things in the initial version of the PNet. We did some filtration for the mutation, remove the silent mutations and the interim mutation, things like that. And the data were ready to be fed into our model. So we made use of the data that is already available for the community to run our secondary analysis. So next, I'll be talking about some computational results. And using a simple standard train test validation split, we were able to come up with good performance. Our model was able to detect like 93% of the primary patients. When we compared our model with other models, including subprotector machine and random forests, we found that our model is has slightly higher performance in terms of the area under curve and the area under precision record curve. When we did the same thing using typical 5-fold cross-validation, we found that PNet is achieving a little bit higher performance as well, with maybe lower variance in terms of the area under curve, area under precision record curve, and also the F1 score. When we tried to compare our PNet to an equivalent dense network, things get a little bit tricky. So if you like to define what's equivalent, you have two scenarios. So you can compare the sparse network to a dense network that has the same number of weights, which means that we will construct a dense network with 70,000 weights, for example, and train this dense network and compare the performance of the sparse and dense network. As you can see from this graph, we trained both of these networks. And when we train these networks using like large number of samples, we find that the PNet is achieving almost the same performance as the dense network. When we decrease the number of samples, we see that PNet is achieving higher performance. You can get up to 20% increase in performance when you have like very small number of samples, like 100 samples. This means that PNet may be useful in the small sample setup, which is the case in the cancer genomics. The second scenario is that you compare the PNet, the sparse network, with a dense network with the same number of nodes. So you fix the number of nodes, however, you have a dense connections between the different layers. And typically, this is a very challenging thing because we have a lot of nodes, especially in the first layer. And connecting this in a dense way is like you will end up with a network that has like so many millions of parameters, probably half billion parameters, and this is like challenging. So we did like something hybrid here. So the network that's called dense here in this graph is a hybrid network where we have the first layer as a sparse layer, and the all of the next layers are dense layer. And you can see that the performance is very comparable. It is both of them are like achieving 0.9 something. So it is like very good performance in general. We tried to see if our model actually generalized to other data that on which this model wasn't trained. And we did external validation, and we got two data sets, one primary samples and one was metastatic samples. And we found that our model is doing good. So the model is correctly classifying 80% of the metastatic patients. We also tried to see how the model is behaving in terms of the errors. So we inspected the wrong classifications of the model, especially the false positives of the model. And we tried to see if the model is doing something reasonable or just doing some mistakes. And we found interestingly that for some of these patients who had like high PNet score, their prognosis was worse. And it seemed that they have higher probability of developing biochemical recurrence after a while. So this gives you some indication that probably PNet is learning something here that is maybe useful and stratifying patients based on their probability of developing biochemical recurrence in the future. So next, in the next few minutes, I'll be talking about the interpretation, and hopefully I'll get to some of the experimental validation quickly. We know that there are a lot of attribution techniques out there that you can use. We used one of the, we used Deep Lift, which was developed at Stanford. And this tool actually gives you the ability to assign scores for different features for each sample. If you aggregate these importance scores on the population level, you can get like an importance score for each feature in your model. So for example, this is the inputs layer. We assigned the scores, and we found that the amplification, the couple number amplification is the most important feature in terms of driving the outcome. You do the same for the second layer, which is the genes layer. You get like something, some genes that are familiar, like AR, TP53, PTEN, RB1, and so on. If you did some factorization and normalization, you can assign also weights for the connections between the two layers. If you repeat this process multiple times, you can get like a holistic visualization of the model itself. And you can observe the flow of the information from the input to the outcome and see what are the most important nodes in each layer. When we inspected the layer of the genes, for example, we found that there are some familiar nodes, familiar genes like AR, TP53, PTEN, and RB1. All of these genes are known in the prostate cancer literature. However, when we went down the list, we found some unexpected genes like MDM4 and FGFR. And while these genes are known to be cancer genes, their role in prostate cancer progression wasn't known before. So we thought that probably there is something interesting here, and we asked why the model is picking on like something like MDM4, for example. When we inspected the next layers, we found that we have a lot of pathways that are related to P53, to the gene TP53. And probably this is why the model actually is picking on MDM4, because it's related to TP53. So TP53 is an important gene. It is involved in regulating a lot of processes inside the cell. It regulates a lot of gene types like cell cycle genes, like death genes, like DNA repair genes. So it is a very important gene. It is tumor suppressor. However, this gene as well is highly regulated one. So there are a lot of ways to regulate TP53. If you are a cancer cell and you would like to stop the function of TP53, you have so many ways to do so. You can mutate the coding region of the TP53 itself, or you can stop the function through different epigenetic mechanisms like phosphorylation or methylation. Or more interestingly, you can amplify things like MDM2 or MDM4, which are known to be negative regulators of TP53. In this way, you can stop TP53 without mutating the gene itself. When we inspected the mutual distribution of multiple genes, MDM4, AR, and TP53, we found there are some genes, like here we have some patients, that, for example, we have 19 patients who have like MDM4 amplification, but they do not have any AR or TP53, for example. So this is an interesting population of the patients. We have also some patients who have like MDM4 and AR. They don't have TP53, for example. So these populations are interesting. Their TP53 is not mutated, rather they have like MDM4 amplified. So we tried to see if there's anything experimentally that can help us make sense of this ranking that we generated. So we went to our friends in Bilhan lab, and we found that they had a ORF screen recently done for a lot of cancer genes. Actually, they screened for all of the genes, like 70,000 genes. And we found that MDM4, for example, here is ranked, it is the highest one that's ranked among the genes that we identified using our model. So the more positive the gene is, the more resistant the cell is for the treatment. So this actually gives us a hypothesis that probably MDM4 is modulating the resistance for the treatment here in the. So we have two lines of evidence now, one from the computational model that we generated and another one from the screen that we got from our friends. We tried to see if we knock down the MDM4 gene, what will happen to these cancer cells. And it seemed that the survival of these cell lines are going down when you stop the function of the MDM4. We also tried to see if we can target this gene using drugs, and we did so. And we found that the sensitivity of such cells actually to treatment goes down when you inhibit such gene, the MDM4, especially in the cancer cell lines that are TP53 wild type. So this was very interesting. I'll summarize here for the sake of time. So we proposed and developed and deployed a sparse biologically informed neural network. This PNet model accurately identified patients with advanced cancer based on their genomic profiles. And we found that PNet was able to recover known biology of advanced prostate cancer, like genes like AR, TP53, RB1, and PTEN, and also discovered MDM4, and it nominated the amplification of MDM4 as important event in the cancer progression, and we showed that this gene can be targeted therapeutically. So in the future, we would like to expand on PNet in terms of the application and also the model development. With this, I'll conclude, and I am happy to get any questions. Thank you.